20th century, genetics had been well established, and we knew that traits were inherited, as Mendel called in these discrete characters, but we didn't know how it happened or what the process was. The discovery of DNA's role as the inheritance molecule is one of the most significant contributions to the understandings of how life works in all of biology. And it wasn't the result of any one person, but the final result of decades of investigations. So we're going to have a quick overview of DNA as a history. First off, you've got Friedrich Miescher in 1869. He was studying pus, white blood cells, and he realized that he was extracting a molecule that was unique from the nuclei, and he called it nuclein for nuclei. Okay, tie that in a few decades later with Thomas Hunt Morgan and his work with fruit flies. Now we know that traits are linked to chromosomes. However, chromosomes are made of this nucleic acid and proteins. So it's not that we're saying that new DNA is the carrier at this point, just that chromosomes carry traits. Okay, Levine in 1909 was an American biochemist, and he was studying these nucleic acid molecules, and he's the first person who figured out the structure of nucleic acids, and he characterized the different forms, DNA from RNA. He found that they contained adenine, guanine, thymine, cytosine, all of those various components. And he thought that these things occurred in equal rates. He called it the tetranucleotide hypothesis, so that A and G and C and T were all equal. And he might have been wrong about that part, but his structural information is still true, and it was very accurate. On to the 20s, and you have Frederick Griffith, and he was studying Streptococcus pneumoniae. He discovered that there were two major types, these smooth colonies that were deadly, and these rough colonies that were harmless. And he found that if he mixed the heat-killed smooth and the normal rough together, the mouse still died, even though both things should have been harmless. And when he extracted the pneumoniae from the cells, he found these living smooth cells. So he called this process transformation, and we know now that that is the process or the capability of the uptake of genetic information from the environment. This was extended in the 40s with Avery, McCarty, and MacLeod, where they took the same test that Griffith did, but each one had all but one thing killed, essentially. Okay, so that okay, all the proteins are destroyed, all the lipids are destroyed, all the carbohydrates are destroyed. Okay, the only one that did not transform was the one that was exposed to a DNase. Okay, the DNA was destroyed. So that showed us that DNA, at least nucleic acids, chromosomes, are genetic information sources. They didn't really take the process to sort of its full extent, but they did say that DNA was the source of transformation, which doesn't really finish the concept, but is the right idea. A variety of other research and experiments that went on in this time period that led to our understanding. Well, one of the more important ones is the work of Chargaff, Erwin Chargaff. He realized that the tetranucleotide hypothesis was incorrect and that there was a relationship, but it wasn't an equal one, that A was to T and G was to C. And you can see those numbers over here on the right. It's not a precise match, but it's pretty close. Now, it's not true for every organism, as you can see in the first line, but that organism, it turns out, is not a double-stranded DNA molecule. So there is actually a reason why it doesn't work. Now, we're going to actually go, remember at this point, DNA is the transforming factor. So in 1952, Hershey and Chase wanted to take this a little bit further. They used the T2 bacteriophage, and they labeled the bacteriophage with radioisotopes. Phosphorus 32 for DNA because phosphorus is in the phosphate group, and sulfur 35 for the proteins because of the cysteine and methionine sulfurs. They exposed the bacteriophage so that they would be radioactive, and then they spun in basically a centrifuge to find out what pieces of information were being passed from the virus to the bacteria. And in the end, they determined that it was only the DNA that was being passed because only the phosphorus was found inside the cell. So at this point, we knew that DNA is the carrier of genetic information. Move that along, that started a global process where they were trying to figure out the structure of DNA. What got there eventually was in 1953, Watson and Crick figured out the structure that we know now as the double helix. They could not have done it without the work of Rosalind Franklin and her labs, which great deal of drama behind, but we'll move on from there right now. The X-ray crystallography images from Rosalind Franklin showed that DNA was a double helix, which was the final piece of information needed to figure out the structure. From that, the next major step was figuring out how DNA replicated. And as they pointed out in their in the actual article where they published their work, was they did not escape their notice about this process because of the weak bond that exists down the center of nucleic acid. It likely replicated, taking that into account. If we wanted to actually look at how we figured out how DNA replicates, that is the Michelson and Stahl experiment. They took bacteria cultures that were with a heavy nitrogen so that the DNA took up a heavy nitrogen that we could separate by weight in a centrifuge. Then they took that bacteria and raised it in a lighter nitrogen so that there was again a weight difference. So there were three major ideas for how replication could work. The first was the conservative model that both strands stayed together, the semi-conservative model, so the two strands split apart down the middle, which was the precursor idea that Watson and Crick were pointing out, or the dispersive model that it was in chunks and somewhat random. When they went through the actual process, when they centrifuged, they found that after one centrifugation, there was only one intermediate band, which knocked the conservative model directly out, because there would have been a heavy band and a light band in the conservative model. When they did the second model, they found two strands, one that was less dense and one that was more dense, meaning that the semi-conservative model was the one that was supported by the data. So overall, we can see that relationship here. One band after first doesn't take the semi-conservative or the dispersive out, but two bands after the first does. So overall, we see that structure of DNA that we know now. You have the major groove at 3.4, you have the minor groove at 1 nanometer, you have the distance between the bases at 0.34 nanometers. These are all measurements that could have been determined from X-ray crystallography images like Rosalind Franklin's. We know that the phosphate groups are in a backbone down the side with the sugars, and the nitrogen bases point in the middle. We know that the nitrogen bases are held together by hydrogen bonds, and that gives us the overall structure of DNA. Your genetic inheritance is part of that structure that you get from your parents. Now, there are different forms of DNA. There are actually three major forms of DNA that we know of at this moment, and it's all about interactions between the base pairs. You have the A form and the B form and the Z form. Most DNA double helices are right-handed, so if you were to hold your hand out with your thumb pointed up and your fingers curled around your thumb would represent the axis. Only one type of DNA, Z-DNA, is left-handed. A-DNA is a rare structural form that happens when DNA is dehydrated. The B-DNA is the more common type, and it's the one that we think of when we think of DNA structure. And then the Z-DNA is a left-handed double helical conformation that has this sort of zigzag pattern. The exact function of Z-DNA is not known, but we do know that it's typically found upstream of the start site of a gene, so it probably plays some role in regulation.